Okay, thanks, Cheryl, and thanks everybody for coming to this presentation. You guys have been hearing about Project 11 Plus for quite some time now, so hopefully this will be a good opportunity to take a bit of a deeper dive on at least my portion of this project, the transcriptomic analysis of the impact of diet composition on gene expression in the black soldier fly larval gut tissue. So we'll go a little bit further into the methodologies used, the way that we approach the data, and what types of things that we found going into it. So as you all know, the research team on the project composes of Heather, Jeff, and Christine, as well as Dr. Federico Hoffman, Dr. Florencia Mayer from Mississippi State, and Dr. Spencer Beamer from the University of, from Texas A&M University. This is also a very large cohort of researchers, including Jordan Smink, Madeline Reeks, Hunter Walt, Brandon Pierce, Sienna McPeak, Aaron Harris, and myself, spanning all three of our major CIF research universities. And then our IAB mentors include Chris Warburton and Pat Crowley, who we're grateful for their time and their input on the project. So overall Project 11 Plus goals are to determine the impact of macronutrient content on life history, traits, gut microbiome, and genetics of the black soldier fly, mealworm, and cricket. That is an umbrella statement to say the least, and we're chipping away at it one day at a time. So this is part of a 3-part seminar series where we're taking a closer look at Project 11 Plus. Last month, hopefully you guys were able to tune into Aaron Harris and Sienna McPeak's presentation on the phenotypic plasticity of black soldier flies, mealworms, and crickets in response to the macronutrients of their diet, focusing on growth and survivorship. Now we are in November, and I will be giving you this presentation. And then next week, Hunter will give a presentation on the responses of the microbial responses of the diet changes in the black soldier fly. So a quick recap, Project 11 Plus is very multifaceted. Last month, Aaron and Sienna presented data on 4 different experiments that we're running as part of Project 11 Plus. The first of which being the Black Soldier Fly lab scale project conducted in falcon tubes with 3 macronutrient ratios, which we'll get into later. The second of which is an extension of this first project with black soldier flies at a larger benchtop scale in shoeboxes, wherein we focus in on 1 macronutrient ratio with or without a microbial inoculation. And we'll get into some more of the details later. And then the last 2 are facets of Project 11 Plus that we won't be talking about here. This is the mealworm and the cricket. While there was samples collected for transcriptomic and microbial analyses, there have been some complications here which have delayed us getting results for mealworm and crickets yet. But today we will by and large be focusing on the black soldier fly lab scale. This will be 95% of this presentation. They'll also be presenting some preliminary data from our black soldier fly benchtop analysis. So again, focusing in on the lab scale experiment. This is a refresher from last week as well, but there were 4 diet treatments originally that went into this project, 3 of which composing of synthetic diet that was produced by the Beamer lab, including a carb-biased, a balanced, and a protein-biased ratio with 1:5, 1:1, and 5:1 protein-carbohydrate ratios respectively. And then we also have a Gainesville control diet that was utilized as a quote-unquote control, though, and we'll get into this later as well, we ended up taking this treatment out of our data analysis, especially when it comes to the transcriptomic work. So the methods for the experiment is the 7-day-old neonates were weighed in groups of 10 and placed in 50 milliliter falcon tubes, 10 larvae per tube. Tubes were monitored daily to evaluate the level of prepupation. Once the tubes were about 40% prepupated, the tubes were harvested. The weights were taken for individual larvae and survivorship was determined. and then samples were set aside for microbial analysis and gene expression. So a quick review of the results from the growth and survivorship data, just to put into context the transcriptomic data that we're about to get into. The balanced diet results in significantly higher larval weight than either of the two biased diet. The protein biased diet had significantly lower survivorship than the balanced diet, and the protein biased diet. The protein biased diet had significantly lower survivorship than the carb biased of the balanced diet. And then the carb biased diet resulted in significantly longer time to reach pupation. This should say carbohydrate, excuse that typo. So then the goals of my research here is to determine the influence of differing protein carbohydrate ratios with artificial diets on differential gene expression in the black soldier fly larval gut tissue. To go over the microbial methodology briefly, for each diet treatment, 3 larvae from 3 random tubes were collected, so an N of 9 per treatment. The cuticles were briefly sterilized using 70% ethanol, and the guts were dissected in RNA later, placed in RNazole for RNA isolation. Quick shout out to Hunter Walt, who slaved away at actually conducting all of this tedious laboratory methodology. So the RNA isolation, again, via RNazole and NEB Monarch RNA cleanup kit, was isolated and purified, and then the purity, concentration, and integrity was assessed using nanodrop, qubit, and tape station systems. Libraries were prepped using NEB Ultra 2 RNA kits. RNA depletion and microRNA or messenger RNA enrichment steps were actually avoided as we were trying to keep all host and microbial reads so that we could conduct transcriptomic analyses on both the larval guts and the microbes themselves. And then it was sequenced using a HiSeq 2000 instrument. So read processing, the reads were trimmed, both the adapter sequences that are necessary for DNA sequencing were trimmed, as well as any low quality reads were also trimmed, and then quality control using the FastQC program ensured that this was all done properly and that our reads looked okay. So then all of our reads were then mapped to the black soldier fly reference genome obtained from CIBI, and a count matrix was generated. And really, this just means that after all of this data processing, we end up with one matrix that contains every single transcript with reads associated with it and how many reads were associated with each of those transcripts. So on average, we had 55 million reads generated per sample. Of that, roughly 54 million reads mapped to the BSF reference. And they mapped to about 15,000 transcripts. 13,500 transcripts were kept for downstream analyses after these reads were filtered. The transcripts were filtered for read count and base mean value just to ensure data quality for downstream analysis. Principal component analysis gives an idea of how much variation in our data sets can be explained by treatment-driven effects. And as you can see, there's a relatively low degree of clustering amongst our different diet treatments, suggesting that much of the variance in our data set is not explained by treatment effects. Not to say that none is explained by treatment effects, however. And just a quick aside that feel free to raise your hand or just pop in with any questions as I'm going through this, because I know that some of these slides can be pretty dense. And so if you have any questions at all, don't hesitate to just jump in. Okay, so then for the differential gene expression analysis, we used a likelihood ratio test, which is a test that identifies significantly differentially expressed transcripts across any 2 diet treatments. And so as opposed to doing pairwise comparisons between each diet treatment, trying to parse the data this way, this is kind of a more holistic approach to analyzing this data. And it does this by creating two models. So one model that excludes the diet as an effect and one model that includes the diet as effect. And then it asks two questions, the first of which being, does the reduced model fit the data worse than the model that includes the diet as a factor? And then if it does, then is this difference significant? And if the answer is yes to both of those questions, then a gene is considered significantly differentially expressed. So through this analysis, we found 401 significantly differentially expressed transcripts, or DETs, and then our BH-adjusted p-value threshold was 0.05. So I'm going to get into the weeds a little bit on a clustering technique that was utilized to analyze this data, just because it's going to come up in my presentation as well as Hunter's next week. But we used a technique called divisive hierarchical clustering analysis, or DIANA for short. This is a common machine learning algorithm. And what it does is it starts by considering all observations into one giant cluster. So you can see that in yellow. You could consider every single transcript, significantly differentially expressed transcript, as a letter in this diagram. And then it systematically bifurcates the cluster to minimize within cluster variance and maximize between cluster variance. And then, well, and so then in this context, each transcript is one of our observations, like I mentioned before, and the similarity between transcripts is based on the expression profile across the diet treatments. And so, for example, here you have a transcript that's downregulated in the low, or in the carb biased diet, upregulated in the protein biased diet, and then the balanced treatment is somewhere in the middle. It uses this pattern to compare these different transcripts. And so for each or expression values from each sample for a given diet are averaged together, and then a distance value is derived from all pairwise comparisons. So for example, on this pairwise comparison, there might be a fairly large distance value generated because this expression pattern across the diet treatments is pretty different from this expression pattern. And then if, for example, this transcript 2 was a more similar pattern, then that distance would be less. And so if you have 4 transcripts to begin with, then the clustering algorithm might split it into 2 clusters because these 2 are more similar and these 2 are more similar. And then it might split again because there's a greater difference in expression patterns here. And it might split again further. And then you're in a position where you could have, if there's 4 differentially expressed transcripts that we're looking at, you could have anywhere from 1 to 4 clusters, right? And so then if there's 401 differentially expressed transcripts, as we had in this data set, you could have anywhere from 1 to 401 clusters. And that the number of clusters, whether it's 1, 2, 3, 4, 5, 6, etc., is determined by minimizing the within cluster variance while maximizing the between cluster variance to figure out how many clusters best represent the data set that we're looking at. So with all of that in mind, these are our cluster results from those 401 significantly differentially expressed transcripts. And they cluster really, really nicely into 4 groups, each one representing a different expression pattern. And real quick, I'm going to go into an aside as to why we excluded the Gainesville control from this analysis. So if you look at just group 1 that consists of transcripts with this general pattern, and then you rerun the same exact program, the same exact clustering analysis with Gainesville included, then all of a sudden this cluster breaks up into a variety of different potential patterns, wherein some of them, the Gainesville is downregulated relative to the synthetic diets, where the synthetic diets are still generally following the same pattern. But then you also see many transcripts that are upregulated relative to this same pattern. And so what this does is it really muddies the water in terms of trying to analyze the data. And this makes sense when you're considering what we're trying to compare, right? There's the synthetic diets consisting of cellulose, sucrose and dextrin, casein, egg albumin, potato peptone, additives and vitamins. And this is, as bare bones of a diet as we can generate so that we can very specifically modulate the protein carbohydrate ratio while changing practically nothing else, compared to a wheat bran cornmeal alfalfa mixture of Gainesville, where there is a huge amount of other variables that might be playing a role, whether it's vitamin and mineral composition or who knows what else. And so while these diets are difficult to compare, these results highlight the complexity of diet-dependent gene expression in the black soldier fly. And we're in talks of trying to create an additional manuscript that takes a closer look at what some of these dynamics might be and what can be gleaned from that information. Okay, so back to the results from the analysis that we did conduct without the Gainesville. You can see that almost 80% of all differentially expressed transcripts found were clustered into one of two groups, group A or group B, both of which, rather than being directionally regulated by the protein carbohydrate ratio, seem positively or negatively correlated with the average larval weight, with the balanced diet producing significantly larger larvae than either of the two biased diets. And within that context, we can look at the functional enrichment analyses. The way that the functional enrichment was done is we use KEGG pathway terms, KEGG orthology terms, and Go biological processes terms. For those who aren't familiar, these are all just different data sets of terms that are associated with genes that give us information about what those genes functionally do. And so by conducting an analysis where you say within this subset of genes, all of these terms are represented, and which of these terms are represented to a significantly greater extent than would be randomly represented across the entire larger data set, rather than just the significantly differentially expressed transcripts, you can see what terms are significantly enriched. And after that enrichment analysis, significantly enriched terms were manually curated to reduce redundancy between terms. And this is really just for ease of interpretation, though obviously the full functional enrichment analyses will not only be in the supplementary material of the paper that we're putting out, but are also available to you guys if you guys are interested. But an example of that is like mitochondrial transport, or mitochondrial transmembrane transport, establishment of protein localization to mitochondria might have all be enriched for one of these groups, and so those might have been condensed to one term of mitochondrial transport. So for this group A, what terms were significantly enriched? Again, these are genes that are upregulated in the balanced diet relative to the biased diets. And fittingly, the largest functional group represented here has to do with developmental regulation. I have highlighted a few of these genes. Heat shock protein, or heat shock 70KDA protein 1 facilitates the proper folding of newly translated or misfolded proteins. PAR3 codes for partitioning. Defective protein 3, which is involved in the establishment of mitotic spindle orientation, gonad development, anterior-posterior differentiation, and metamorphosis. And a lot of these developmental genes specifically had to do with things like dorsal ventral pattern formation, protein localization, imaginal disc formation, so preparation preparation for metamorphosis. And so all of this goes to show that when these larvae are getting the nutritional inputs that they require, more energy can be devoted to, whether it's preparing for metamorphosis or just normal growth and development. So all of this is fairly unsurprising. Group B, which is the inverse of group A, genes that are downregulated in the balanced diet relative to the biased diets. these terms are things like biosynthesis, electron transport chain, fatty acid metabolism, lysozymes, mitochondrial transport, which suggests that larvae in micronutrient poor environments upregulate genes necessary to mobilize and utilize the lipid resources that we have, right? So many of this has to do with the metabolizing has to do with metabolizing lipids and converting those lipid stores to energy, which makes sense as to why when these genes are upregulated, the larvae are smaller. A few specific examples of genes represented in this group include TIM44, which mediates translocation of proteins into the mitochondria, ELO, which is the long-chain fatty acid elongase involved in the determination of adult lifespan, lipid metabolic process, and regulation of gene expression, and EC1, which plays a role in fatty acid beta oxidation, protecting against high-fat diet-induced stress in mice, which I thought was interesting. But again, these terms that are enriched make a whole lot of sense when you look at it with respect to the larval weight outcomes that we saw in the phenotypic data. So then moving on to group C and D, these are differentially expressed transcripts that were positively or negatively correlated with the protein carbohydrate ratio in a really surprisingly linear fashion. Group C, so here expression increases as protein increases or as carbohydrates decrease. Here we see transcripts to do with proteolysis, amino acid synthesis, and translation. Specifically, there's an aminotrans-4 gene represented here, which catalyzes the catabolism of branched-chain amino acids. Trypsin, which is widely considered one of the most important digestive proteases in most insects. And RIO1, which promotes ribosome synthesis and subsequent protein synthesis. So as protein increases, these larvae are transcribing the molecular machinery necessary to not only digest these proteins, but also to recycle them into new proteins. And then looking at group D, which again is the inverse of group C. This group had the least amount of transcripts, but also some of the most interesting findings. So these genes had expression increase as protein decreased or as carb increased. And there were 5 different copies of lysozyme coding genes. And lysozymes in black soldier flies are not only antimicrobial peptides, which hydrolyze the peptidoglycan of bacterial cell walls, but also have been found to act in a digestive capacity in mites, enabling the utilization of bacteria as a food source. And this has also been shown in a couple of flies. I know the house flies have some evidence for lysozymes acting in a digestive capacity. Also, there's a lot of study in ruminants and strangely in rabbits around lysozymes for digestion. But this is the first evidence that I've seen that black soldier flies are also utilizing these antimicrobial peptides, these lysozyme genes, as means to digest and sequester resources from the bacteria in their diet under dietary stressors. And interesting too, I'll go back quickly. You see more lysozyme genes here, and these are different lysozyme genes that are upregulated in group B, suggesting that lysozymes, maybe it's not necessarily just a protein-dependent interaction here, that these lysozymes are also, or some lysozymes are also playing a role in sequestering resources from bacteria when there's not enough carbohydrates in their diet. So all this to say, these lysozymes seem to be quite important for better utilizing a wide array of different protein carbohydrate ratios in their diet. So some closing thoughts here is that no study to date has investigated effects of such specific protein-carb diet modulation on black soldier fly transcriptome. That said, there are other transcriptomic studies looking at black soldier flies fed a variety of different diets. For example, this is a study that looked at standard diet versus vegetable mix. There are others that include various additives to the diet. But the importance of breaking this diet down to, as a synthetic diet where we can really precisely modulate the protein and carbohydrate ratio, and only that is super important, especially given the complex effects that we've seen with the Gainesville diet relative to these synthetic diets. So while black soldier fly gut tissue is adaptable to a wide range of substrates, in our study, we saw a relatively small fraction of our differentially expressed transcripts that had a directional relationship with the protein carbohydrate ratio, right? With most of our differentially expressed transcripts having that kind of parabolic relationship where it seemed to be more of a response to general diet related stress rather than a directional relationship with the protein carb ratio. And so with that said, the protein carbohydrate specific responses were limited, which is similar to work done with other insect models. This is a paper that looked at caterpillars and found similar results. And they also found here that this phenomenon is compensated by constitutive expression of digestive enzymes. So this might not be modulation based on the diet that they're eating, but rather they're always expressing genes that can digest kind of the whole gambit of protein to carb ratio diets. And then also microbial interactions. And we'll obviously get much more into that next month when Hendra comes on to talk about what he's found. But I also do want to chat briefly about the black soldier fly benchtop data. So here we have only two different treatment groups. So we're focusing in only on the artificial diet with the 1-to-1 protein carbohydrate ratio, but one of these two diets was inoculated with Klebsiella and one was not inoculated with Klebsiella. Klebsiella was chosen because it was found to be enriched naturally in the black soldier fly larval gut. So it's not necessarily a pathogen. It's theoretically a bacteria that is already there and playing a significant role in the black soldier fly gut. So again, 7-day-old neonates were placed on inoculated versus non-inoculated dietary treatments in shoeboxes this time with 1,000 larvae per shoebox. To get average weight across treatments each day, the 10 largest larvae from each shoebox were weighed, then returned to the substrate. 3 consecutive days of no change or drop resulted in the harvest of the shoebox and the larvae. The total larval weight was taken, survivorship was taken, and molecular methods were described, were the same as described previously. As for both the weight and survivorship data, thanks to Aaron and Sienna for collecting this data, there was no significant data found in either of these two metrics, weight or survivorship, across the two diet treatments. And then very surprisingly, so the number of reads that were sequenced was very similar to what we have found in the previous small scale study. But here we found a shockingly low number of transcripts that were significantly dysregulated between the two groups. So there's very, very little response from this bacterial inoculation. The principal component analysis revealed moderate clustering between the samples of the same treatment. Much better clustering, actually, than we saw in the previous experiment. Virtually no significant GO or KEG terms were significantly enriched amongst the 52 differentially expressed transcripts, which really isn't surprising given that there were only 52 of them. But of note, one lysozyme transcript was upregulated in the bacterial enriched group, again suggesting that these lysozymes are likely playing a very significant role in responses to different diet treatments in the larval gut tissue. And this lysozyme could either be associated with immune response to this bacterium or acting as in a digestive capacity, as we talked about earlier. So some closing thoughts here. The black soldier fly are extremely resistant to changes in bacterial communities of their diet. This is something that we've known for a long time, and specifically in this case, we're really only looking at one bacteria that again already existed in the black soldier fly gut, and saw very little transcriptomic response to that enrichment. So some closing thoughts is that anytime you're working with such large data sets, there are always a variety of different approaches that you can take to trying to analyze it. And in that respect, we really only scratched the surface of what might be able to be gleaned from these large data sets. And so with that being said, if any IAB members are curious about specific genes, whether we saw significant dysregulation in any of our Project 11 studies, past, present, or future, reach out and let any of us know. We're happy to help, and we're happy to do more focused analysis if you want to look at very specific genes, or if you want access to raw data for your own internal analysis, just reach out, and we're happy to provide this data to you as well. So next up in this seminar series, newly anointed Dr. Hunter Walt will tie a bow on the Project 11 seminar series with his presentation on the black soldier flies arming, microbial response to changes in diet composition. And we have manuscripts plural in preparation for Project 11 Plus. So keep an eye out for the write-up associated with this data, last month's data, and next month's data.